it's on. You guys hear me? A little higher? Is that better? Okay. So, a true story, actually, a little bit of my history in terms of the connection to this organization. I about almost 20 years ago, I guess, finished the first year of clinical training in my fellowship at Johns Hopkins. I was a pediatric oncology fellow, and I sort of ended up in oncology with a strong interest in immunology, and I had this idea at the time. Others were thinking about it, but many people thought we were crazy that we could potentially use the immune system to treat cancer, and I was specifically interested in using that approach to treat leukemia. And when I was looking for a lab, I actually lived in Bethesda. My wife was actually working in DC, and so I was commuting back and forth to Hopkins, except when I slept in a dorm room across the street from the hospital frequently as a first-year fellow. And I ended up being given permission to look for labs at the NIH, and I ended up working with Crystal Makel, who is also a CCF award recipient, currently now out in Stanford leading their cell therapy program. And when I went into Crystal's lab, I said, listen, I want to, she was an immunologist, had a strong immunology lab, and I said, I wanted to use immunotherapy to treat leukemia, and she said, listen, you can come learn immunology here, but I'm a solid tumor person, you're going to have to deal with solid tumors. I said, fine. So she was a great mentor. I learned a lot about immunology, and fast forward about 5 years, I was sort of in the postdoctoral training phase of my fellowship, and had done a fair amount of immunology work at the time, developed mouse models, and I decided it was time to start thinking back about the leukemia part of this. And her lab wasn't really funded to do that, wasn't focused on doing that. And so I actually wrote a grant at the time to sort of transition the work I was doing with immunology from solid tumors into leukemias. And that was actually a grant that I wrote to this organization. And so they actually funded the transition literally from being in a mentored lab with Crystal working on immunotherapy of solid tumors and into a focus in immunology that I sort of have carried through to this day. So that's a true story, and is really a testament to the importance of this organization. It's a difficult time, that transition from being in a lab, sort of in a mentored environment, a lot of enthusiasm, a lot of great ideas, and trying to figure out how to take those sort of to the next step as an independent investigator, and that's a vulnerable time too. And so I really think that that's one of the critical functions of foundations such as this. So, true story. So I'm going to talk about the overview part of it will certainly be in this, but I'm going to tell a story about sort of pretty remarkable success that has taken place over the last 5 years or so. Focused on using genetically modified T cells to treat leukemia, pediatric leukemia. And there's a couple of interesting parts of this story that I'll get back to along the way. One is certainly the fact that the results of the very early clinical trials were pretty remarkable, not something that we typically see. The second thing that I'm going to talk about a little bit is that this is a remarkable, unusual example of a situation where most of the first-in-human trials, and I'll get back to this, the first clinical trials with this approach were actually done in pediatric leukemia, not in adult cancers. And so the adult groups have now adopted this and are now running phase 2 clinical trials in adult cancer patients. But this therapy was really, truly developed in its infancy in pediatric cancers. And so it's a really I believe, hopefully most others believe, that there's a role for that. I mean, often we get hand-me-down therapies, if you will, in pediatrics from the adult world, and this was really a case where it was developed fully in pediatrics. So what I focus on in the lab, or what we've been focused on in the lab, has been immunotherapy for a type of leukemia referred to as ALL, acute lymphoblastic leukemia. The most common diagnosis, single diagnosis in pediatric oncology. It's really been an area where there is really the poster child for the development of multi-agent chemotherapy. I mean, if you go back into the 60s, I mean, this was a uniformly lethal, disease, and currently, 85, 90% of children that are diagnosed with ALL will ultimately be cured, and that means, alive without leukemia in 5 years. One of the things, though, that's been interesting and really sort of disheartening with this, and this has been the case with many other cancers, is that, that's great for the patients who do well, but that still means that there's a large subset of patients that won't do well, that ultimately relapse. And really, there hasn't been much progress in that area. And so this is sort of data from a publication that came out of the Children's Oncology Group a few years ago. And what it looks at is the ability for these patients who relapse with ALL to achieve a second remission. And what you find is that, if they relapse once, 85% of those patients will go back in remission number 2. If they relapse again, it'll be 44%, and as you can see, it drops off, sequentially. And even more important is if you look carefully at survival in these patients who relapse, it's really pretty dismal. The CR means complete response or second remission, complete remission to a second attempt at induction. And what you see there is that first time, remember I'm saying that first time when they're first diagnosed, 85% will be cured. Even in that 85% that achieve second remission, the survival is still pretty dismal, and it drops off progressively since then. And again, if you look at these sorts of curves 10 years ago, they're not any different. 15 years ago, they're not really any different. There's been very little progress in this group. The end result is that leukemia still remains a leading cause of cancer-related mortality in children. The other thing that's important is that success, 85%, is really has not been seen in adolescents and young adults. There's been improvement if you apply so-called pediatric inspired, I love this term, the adult oncologists call it pediatric inspired treatment regimens to this young adult population. They do better, but they still don't do as well as younger children. And we all know about the fact that these patients suffer pretty substantial treatment-related morbidity. So what I'm going to talk about today is using something called, these genetically modified T cells to treat this type of leukemia. And it takes advantage of a couple of interesting and important aspects of the immune system that have been used independently to treat cancer. So everybody sort of, I think, knows that you can use antibodies that can be targeted against any protein, your favorite protein on the surface of a cancer cell, Herceptin, rituximab. These are all antibodies that target proteins on the surface of cancer, obviously. And you can manufacture just about anything you want, an antibody target just about anything you want. One of the problems with this approach is, although it can be effective, the killing of a tumor really requires engagement of another immune effector cell or linkage to some sort of a toxin that will ultimately result in death of the cancer cell. The other component of the immune system that can be used effectively to treat cancer is something referred to as a T cell, a T lymphocyte. And we know that these cells, when they see the cancer, can very potently kill cancer cells, and they do it without any other immune effectors. They can do that on their own. That's what they were evolved to do, although they're evolved to do this really in the context typically of infection, viral infection, et cetera. And one of the things that happens is that through a very complicated process, when these cells develop in the body, they become tolerant to yourself. I mean, that's really, otherwise, the process of autoimmunity ensues. And so one of the challenges in using T cells to treat cancer is trying to break tolerance to self when you try to get them to react to a tumor. And so what we're going to talk about today is something referred to as a chimeric antigen receptor. And what these receptors really are chimeric receptors that utilize the antibody. Got it. They use the antibody binding domain in the extracellular portion, and then they link that to a T cell signaling domain. And so you end up with what's referred to as a chimeric receptor, because it contains the antibody portion and then the T cell receptor portion. And then you can genetically engineer a T cell to express this receptor, and in that way it's re-educated to recognize a different protein than it was otherwise developed to do. And so this is what you end up with, is this what's called a CAR T cell that has this construct on the surface. And it combines the target specificity of an antibody with the potent cancer-killing ability of a T cell. And this allows us to essentially choose targets without relying on the immune system to determine what those targets are. And so when we treat patients with this particular therapy, the way this works, is this the pointer in the middle? Yeah, there is. The way this works is that we collect these T cells, non-engineered obviously, from these patients. And then these T cells are expanded in culture in the laboratory, in a clinical laboratory, in the presence of these beads that express these stimulatory antibodies in the surface targeting the anti-CD3 and anti-CD28. And then we use these vectors, viral vectors, actually some of them based on the HIV virus, remarkably, lentiviral vectors, to engineer these cells to express these chimeric antigen receptors. We could then grow these cells up, and then these are reinfused into the patients. And this is a publication that came out of our branch, the clinical trial led by Trey Lee. Crystal Makel was the senior author, using one of these chimeric antigen receptors that targets a protein on the surface of acute lymphoblastic leukemia referred to as CD19. And this was one of a number of first-in-human clinical trials, phase 1 clinical trials, that was conducted in our branch. But I will tell you that there were multiple trials, and I'll show you a table of some of these that were being conducted simultaneously at University of Pennsylvania, Sloan Kettering, Seattle. And this is what's called a waterfall plot. And each of these bars on this plot represents an individual patient. And it shows percent change in the amount of leukemia, the blasts in their bone marrow, from the time that they were enrolled on the clinical trial to the first evaluation at a month. And minus 100 means minus 100%, meaning all the leukemia is gone. And so in this trial, the ultimate complete remission rate in these patients was 60%. These were all chemotherapy refractory patients that had failed, for whom all standard treatments had failed. Most of these patients had undergone a bone marrow transplant prior to enrollment on this clinical trial. In general, we think of a promising phase 1 clinical trial result to be 20-30% response rate. This is remission rate in these patients. It's really remarkable. And again, all of these were children enrolled on this clinical trial. The other thing I'll point out, just as I know, many of you in the room are probably aware of this therapy is not without toxicity. The primary toxicity is something referred to as cytokine release syndrome, or CRS. And what you see is all of these hashes are patients that develop some degree of cytokine release syndrome. And it's been observed across a number of clinical trials. I'll tell you, this trial, this is 20 patients. We ended up treating over 50 patients on this trial. The response rate held up. There have been well over 200 patients probably treated nationally now with CD19 CARs, probably more now. And one of the things that we've seen is that the development of cytokine release syndrome is linked to the amount of leukemia that these patients have, and it correlates with the likelihood that these patients will ultimately have a response. So the toxicity definitely seems to correlate with the success of this approach. So this is a table of clinical trials. This was a few years ago, a supplement that Crystal and I published at the American Society of Hematology meetings, 2013. And it was a table of the ongoing trials at the time. I'll point out that much of this work has been very rapid. Much of this has happened in the last 3 to 5 years. These were ongoing trials at the time. And what I'll highlight is that, whoops, how fast. Every one of these trials were pediatric trials. These were all the early phase 1 clinical trials, and all of those were pediatric trials. So as I said, at the beginning of my talk, this is really an example of a place where this therapy was really developed in the context of pediatric malignancy. So, with success, we learn. And one of the things that has been apparent from CAR T-cell therapy is that, and everybody who does cancer research in the room knows this, is that tumor cells are pretty remarkable in their ability to figure out ways to evade therapeutic approaches, and that has certainly been the case here. what we've seen is that with this type of therapy, at least 10%, the last data from the University of Pennsylvania with persistent CAR T cells is that it could be as high as 30+% of patients after some period of time of being in remission, these patients will relapse with leukemia that loses expression of the CD19 protein on the surface, at least the epitope that's targeted by the CAR, the antibody binding portion of the CAR. One of the things that we were interested in the branch, and actually we started this work prior to this observation of CD19 loss in the context of these therapies. I'll point out as an aside, there's another agent called blinatumomab that's a CD19, CD3 bispecific agent, and the same thing is being seen with blinatumomab. The incidence seems to be maybe a little lower. It may speak to the durability of the pressure on CD19 with CAR T cells, but it's certainly something that we've seen. But one of the things that we were interested in the branch, based on some of what we were seeing, the success we were seeing in the clinical trial, was could we take this and figure out whether we could come up with alternative CARs that could be used successfully. We developed a CAR in the pediatric oncology branch targeting another protein that's expressed on the surface of pediatric leukemias, referred to as CD22. And there had been a number of studies done, and Dr. Shaw will talk, I think, in a little bit about antibody approaches targeting CD22. And I'm not going to go into all the work. I will tell you that making a new CAR is not necessarily trivial. You can take about 15 different antibodies that bind very effectively to the protein, and none of them will work as a chimeric antigen receptor. We're just learning the rules about how to make a good chimeric antigen receptor. But ultimately, we came up with one that seemed to work and was published in 2013 in Blood. With that particular CD22 CAR, I opened a clinical trial in the pediatric oncology branch that was sort of modeled after our CD19 CAR experience. And it was sort of a classic phase 1 clinical trial, whatever that means, with a cell therapy that is really a living therapy that expands after infusion into the patients. I mean, this is the model for drug development. It's very different when you're doing this. But what we ended up doing was starting with a dose that was a half a log lower than what was active in the CD19 CAR experience, 1 × 10⁶. And the process was that we would enroll patients in this trial. We would give them chemotherapy, which seems to be important to prepare them for infusion of the CAR T cells, and then evaluate for response at about day 28. The plan eventually is to pick a dose and expand to look at 2 specific groups, patients. This is really the first trial where not only we've tested a new CAR beyond CD19, but also one where we have an experience treating patients that had received another CAR. So a lot of the patients that we've enrolled got CD19 CAR, failed CD19 CAR, many of them because they lost antigen, and then treated them with another CAR. And so we plan to stratify based on whether or not they had received previous CAR. So I'm not going to go in this table in great detail, but these were the preliminary results so far. We've actually treated an additional 3 patients that are really too early to speak too much about. But the first dose level was 3 × 10⁵ per kilogram. And in this cohort of patients, we had 1 patient who achieved a complete remission. This was a patient that got CD19 CAR, lost CD19 expression, went into remission, and then subsequently relapsed at 3 months. But at least gave us some evidence of activity. We definitely saw cytokine release syndrome. I'll point out that all of these patients had received a prior transplant, one of them 2 transplants, the patients who achieved remission. A number of these patients had received prior CD19 CAR. We then dose escalated to 1 × 10⁶ per kilo. This is the active dose in the CD19 CAR experience. Treated 3 patients, and every one of these patients achieved MRD negative remission that was sustained beyond 3 months. So really remarkable for a first-in-human clinical trial. I'll point out that one of these patients was a patient that had received prior CD19 CAR. At 1 year after CD19 CAR, she relapsed with CD19 negative leukemia, got the CD22 CAR, entered remission, and she'll be coming back at 6 months for her evaluation. She's graduating from college in a few weeks. So really a pretty remarkable experience so far with this trial. It looks quite promising. So, this is this patient that I mentioned who had received prior CD19 CAR, patient number 9 in the clinical trial. This is what her leukemia looked like at enrollment on the clinical trial, defined by expression of CD10. And as I said, although leukemia should be here, positive for CD19, hers had lost CD19 expression. She retained CD22. I'll tell you, the density or the expression level of CD22 is pretty low, and we weren't quite sure what that was going to mean. But this is what happened after we infused the CAR T cells. The top here shows her leukemia in her blood at day 1. This is all leukemia in this gate right here. This is clearance of the leukemia by a month, and this is expansion of the CAR T cells. No CAR T cells at the beginning. 22% of her T cells were CAR positive, and this was sustained out through a month. I'll tell you, she had persistence of her CAR T cells through at least 3 months, and we'll see where she's at 6 months. So, I think sequential therapy seems to work. Our general impression is that this is probably not the best way to go about doing this. We think that probably you've got 2 good CARs. That, we know that that combination therapy seems to be effective with chemotherapy, so we're certainly thinking about how to combine the CD19 and the CD22 CAR. We've actually built a CAR in the lab that has both binding domains in the same construct. It's active. We're able to clear leukemia. We're making vector right now, and we will hopefully have this clinical trial open in about 6 to 12 months here and at Stanford. So I'm just going to very quickly, how am I doing on time? I got to finish up here, don't I? Got a few minutes. Yeah. So I'm going to actually skip through this, but I'll just tell you that we've been interested, obviously, in thinking about other targets on other types of leukemia. One of the things that we've done is to focus on a subset of leukemias, a rare subset, infant leukemia. Not a lot of enthusiasm for this, in the pharmaceutical world, just because it's a rare population, but these patients with infant leukemia do dismally. We're also interested in AML, and we know of a protein, FLT3, that's expressed on the surface of both of these types of cancers. We've made a FLT3 CAR that is quite active. This is in a FLT3 positive leukemia. This is looking at all the red and the blue are leukemia. This is no CAR treatment. This is different doses of CAR T cells. This CAR is quite active, and we're able to clear essentially an infant leukemia in this mouse model. So we're quite pleased with this result and planning to take this forward. I'm going to stop now. I'm not going to get into sort of the work that we're doing in the lab, but I want a couple of things. The general themes right now, I think that's emerged from this field is that we know the primary toxicity is cytokine release syndrome. It's not trivial in some patients. Many of these patients can land in the intensive care unit. It's associated with elevations in multiple cytokines. We know that it correlates with the amount of leukemia these patients have at the time of CAR infusion, but it also correlates with response. And so there's a lot of interest in trying to understand how to sort of manage and balance these, 2 different aspects of cytokine release syndrome. An incredible thing of this with this therapy is compared to a lot of other adoptive cell therapies for cancer, the dose that we're giving these patients is tiny. We're giving these patients on the order of about 50 million CAR T cells. In a lot of immunotherapy protocols, billions of cells are required to get regression. And so that requires huge expansion of these T cells. And a lot of what we've done in these clinical trials is to assume that these cells behave just like a normal T cell. I can tell you from our mouse models that they don't, and there's still a lot of biology that we have to sort of understand to try to understand whether or not the patients who don't respond are really having problems with this issue. In patients who have a response that fail where the leukemia comes back, in some of those patients they lose antigen. There's other patients where it's a T cell failure. And when we retreat those patients, they don't respond to the second infusions as well. We don't understand that, but that's obviously something that's important. I don't think this is going to be a bridge to other therapies. I think the ultimate goal with this, given the success, is to turn this into a durable therapy. And I think, in order to do that, we're really going to have to understand, I think, how to get some persistence of these CAR T cells. We're reasonably good at that, but that's not true in all patients. And I think ultimately, if we want to turn this into a definitive treatment, I think that's something we have to understand. So I'm going to blow through this part. I'm just going to say that what we've done in the lab is to sort of turn back to some of our basic immunology models and try to apply murine models of CAR T cells to understand this biology, because a lot of the models that have been used to date are these human xenografts They're highly immunodeficient mice. They lack a lot of the complexity that's required to understand some of this biology. And so we're really behind in that area, I think. And I think it's a place where we can certainly optimize this. So I'm going to, with that, stop and acknowledge, I certainly acknowledge this foundation at the beginning for enabling me to transition from solid tumors into leukemias. The work that I showed was conducted by Haiying Qin, Amy Yang, did a lot of the preclinical modeling that I showed. Haiying made the FLT3 CAR initially. Chris Chen has sort of developed it since then. These are the folks in the branch that did the work. And Raleigh, actually, who was over here, who might have moved, is the clinical lead in the CD22 CAR trial. I take the credit. She does all the work. That's not fair, but she really is. It's a huge effort to get these patients enrolled, and I really owe her a lot in that regard. And we make these cells in the Department of Transfusion Medicine. This is an individualized product. It takes about a week to make these. It's a fair amount of work. Patrick may speak a bit about aspects of cell production in his talk. Finally, the CD19 CAR was developed in the surgery branch, and obviously very indebted to patients and their families. So with that, I'm going to stop and turn it over to our next speaker, who is Patrick, right? Okay. Sure, sorry, I didn't want to, go ahead. Yeah. Trey and Crystal, in all fairness. I was on the trial, but Trey and Crystal.
So, you can publish this great data and other people, but you know for a fact that all their patients were not treated in the best way, or they were able to make cell products.
Yeah.
So here you are proposing to make it even more complicated cell products.
The 1922 bispecific, yeah. Yeah. I mean, I think that you have to do it as an intent to treat, with the understanding that what you're going to do is you have to look carefully at the patients who don't respond, the patients who you don't make products for. You have to understand that. You always put stopping rules, as you know, in the trial. If you can't make products in the first 3 patients, you got to go back to the drawing board and understand that. Many of the patients, I mean, what you speak to is the fact that a lot of the patients that were enrolled on the other, the Penn trials and others, what's done in those patients is that there's often a test expansion where they try to expand a very small number of T cells. And if they can't expand the T cells in culture, they don't enroll them on the trial. And then you end up with an 80-90% response rate in the patients for whom you can expand their T cells. So that's great. It tells you if you can treat a patient that it's a really effective therapy. But what we want to understand, I think we, can do, with some of these more basic questions, is to go back and look at the patients who, it's very effective, clearly. There's no reason to hide behind a 60% complete remission rate in a first-in-human clinical trial. There's nothing to apologize for. It's pretty remarkable. But I want to know why the 40% of patients who don't respond, and I want to understand the patients who relapse, why they relapse. That's really, I think, what the trick is at this point. So to be honest, I think, is the only way to approach this. But we'll see. Yeah. Yeah. Wnt. Yeah. Yeah. It hasn't been, but it's being, I should say it's being explored preclinically. It hasn't been done clinically yet. So Luca Gattinoni, who was with Nick, is running his own lab in ETIB with Ron Gress. And he had come up with some microRNAs that could essentially make TSCM, stem-like T cells. And so he's using those microRNAs, in the context of CAR trials to try to see if he can improve responses. So definitely being thought about, and I think very soon we'll start seeing trials taking that approach.